Hi, I'm Sydney Herford, and this is my protein presentation. My protein is histones, and let's start. So what is a protein? Proteins are macromolecules. They function in a variety of different things, and so the building blocks of these macromolecules are the 20 amino acids that you see here, which combine and make up protein, and they combine in different ways to form their unique protein. Okay, and then we're going to go on to protein structure. So all studied proteins are comprised of one or more structural configurations. So we have primary, secondary, tertiary, and quaternary. So the primary structure is defined by the specific sequence of amino acids which make up the polypeptide chain, and assembly of those is achieved via a series of dehydration reactions. And then the secondary structure is determined by how the polypeptide's backbone is formed, or folds, And so that can either be an A helix, which is the alpha helix you see over here, or the B pleated sheets, which is what you see over here. And then additional interactions between the varying side chains will determine the tertiary structure, which is the three-dimensional structure. And lastly, more elaborate proteins like hemoglobin combine multiple subunits to form a final quaternary structure, which is down here. Okay. And then, so now we get to histones, and what are they? So what I put here is a family of proteins responsible for condensing DNA so that it can fit within the nucleus of a cell and that they're positively charged. So the negatively charged phosphate that makes up the backbone of a double helix is associated with the positively charged histone, and that's how it is able to condense and fit into the nucleus. The DNA essentially wraps around the histone and condenses to form beads, which are nucleosomes, but we will get there. Okay, so right here I have the H3, H4, H2A, and H2B, and this is the primary structure of histones up here. And these are typically composed of amino acids like arginine, histidine, and lysine, and these amino acids are what allows them to bind to the negatively charged phosphate and the DNA backbone. And then the secondary structure also share a commonality, which is a domain called the histone fold. The histone fold is composed primarily of three alpha helixes. You can see those over there. And they all come together. So they make loops, and these loops are extremely significant, and they allow for the heterodimeric interactions between the H3, H4, H2A, H2B, the core histones. And heterodimer formation of cores H3 and H4 further facilitate the formation of tetramers. And then lastly, we get to the octamer, And this is the final quaternary structure of histones. Hold on, let me think. And then, once a lot of these are formed, they will, one of those will form, and you'll get a nucleosome, and the nucleosomes will interact, and you will eventually get, so here's another nucleosome, and here's the ribbon model of the H3, H4, H2A, H2B. You'll eventually get the, what's it called? I'm sorry. Chromosome. That's what it is. Sorry, I blanked. Alright, sorry, I got off track a little bit. So these complex proteins that serve a variety of functions in eukaryotic cells. DNA packaging and regulation of gene expression are just a few of the vitally important functions that histones serve. And more from an evolutionary perspective, these proteins have facilitated the development of more complex organisms by increasing the amount of information that can be efficiently stored within a cell. And without histones, it would be highly improbable that the transition from prokaryotic life would have ever occurred, because of its elaborate mechanisms, which enable complex life to exist in the first place. And that is my presentation. Thank you.